Good day and welcome to Mr. Wilson Live. It is another Friday evening and we are here to do the business of CXC. The biology and HSB syllabus is what we're looking at. And this evening we're going to be looking at inheritance. For the past couple of 2 weeks, I think, the first week we did past papers, and last week I gave you an opportunity to watch some videos on inheritance and, of course, variation. Now, this week we are going to be doing the practical, and I'm supposed to be here with you for at least an hour. So I encourage us to get ourselves pen and pencil, and we're going to be needing book and possibly paper. So we're going to need those for class this evening because the class is going to be all practical, as I will pull you closer to that which you hate to do. But at the end of the lesson, I am sure that you'll be much more comfortable and you'll be able to solve the questions that require you using the Punnett Square or the genetic diagram, whichever you'll be more comfortable. Now, if you have not yet liked, share, or subscribe, it is definitely time for you to do so. I don't know if I'm echoing where you are, but I know that I'm not wearing that earphone. But for some reason, I feel a little more comfortable not wearing the earphone. So for that reason, I'm not wearing the earphone, and I'm hoping that it's not too bad where you are. If it is, I'll just put on the earphone. So if you have not yet liked, share, or subscribe, it is definitely time to do so now. And if you have not invited a friend to the show as yet, it is still a good time to do so. I'm seeing Miller here. I think Miller is new. I don't know that I know the name Miller. Good evening. Are you new, Miller? All right, we're going to be kicking off the show with inheritance of sex. That's a very easy diagram, and I'm going to be doing that. And then I'm going to be running through a couple of other diagrams, but I'm not going to be leaving you. I'm going to go through very slowly. So while we wait on the others to join, let me just do my roll call. And having done the roll call, then we will hop right back into the lesson. So Barnett is with us. Good evening. Powell is with us. Good evening. Moore is also with us. Good evening. Martin is with us. And of course, Cheryl is with us, and Miller is with us as well. We are going to be starting off, and I'm going to be showing you pretty much how to draw that genetic diagram. Do you have a pen or a pencil, book or paper? Just let me know in the chat. Just say yes, and then we can start the game. All right, do we have pens and paper? Are we ready now? This is more than just a lecture session. This is a practical session. Pen and papers, tell me yes in the chat so I can begin. All right, yes, sir. Yes, sir. Beautiful, beautiful. All right, just give me a second. I think I'm having too much reflection behind me. All right, I think that might be a little better. Good, thank you. It's my second time, but I watch almost all your videos. Beautiful. We're very happy to have you. Please ensure that you subscribe. All right, I'm going to show you the easiest thing first. If you have watched IDD, if you have watched my multiple choice papers, you would have seen the genetic diagrams, and you would have seen me working it, and sometimes it's not so pretty, but I'm working it on the paper because you want to solve it very quickly and move on. So the first thing we need to do is to learn how to draw that simple diagram. In the exam, if it's a multiple choice, we don't need to get into the detail, just solve the question. So first I have a blank sheet of paper. I hope you can see it well. So the first thing I want you to do is to make a nice size rectangle. So that's the first thing you're going to do. Can you see my nice size rectangle? Beautiful. You would have seen this in the videos. Then I want you to make two horizontal lines. Two horizontal lines. So it's going to look like this. All right, so the diagram looks pretty much like that. And then I want you to make two vertical lines. So the diagram looks pretty much like this. All right, so the first thing what we're doing now, it is called a Punnett Square. So this is pretty much a construction of a Punnett Square. So we'll make a rectangle. It's the easiest way for me. I've seen other diagrams, but it's the easiest one for me. So I'm going to go with the easiest one at all times. So we made a rectangle or a square, some might say. Then we drew two lines horizontal. Then we drew 2 lines vertical, and then we're going to now just draw a line diagonal in the first cell. So if you observe here, the first cell is diagonal. Now we might choose to put something in the first cell, probably. Or probably we just leave it as a cell that we're not going to use. Now, are we there as yet? Everybody all right with the Punnett Square? Yes, no? Are you okay? Are we all right with the Punnett Square? Yes, no? Tell me in the chat, guys. I am not leaving you. Tell me in the chat. Are you fine? Yes. All right. So everybody learn to construct the diagram. All right. So I like to get that part out of the way. You draw a rectangle or a square, and then you draw 2 horizontal lines, 2 vertical lines, and then a diagonal line in the first cell. Punnett square. That's the first thing. Beautiful. Now, we're going to use the square to do almost all the crosses that we're going to do. But we must bear in mind that all the crosses that we're going to do, they are going to be different. So you have to follow me that as soon as I change the rule, you are there with me. If you are not following when the rules are being changed, then you are going to become confused and you might fall out. We don't want anybody to fall out. So being that we have done the first step, I'm generating the Punnett square, I'm going to get another blank paper. And what we're going to do is to do what we call sex determination, or how we acquire sex. They tend to do that. YouTube at one time had blocked me for that because the software didn't understand what we're talking about. Now, what we're trying to figure out here, I think the family with most children in the world is from Germany or somewhere there. I really don't remember. But what we want to find out with the determination of sex or inheritance of sex, we want to find out if you were supposed to have a child under normal condition, what would the offspring be the next time? Even if you have 99 girls, the next time that you happen to become pregnant, would you have a boy or will it be a girl? That's what we want to find out when we talk about sex determination or inheritance of sex. So the first thing we want to do in answering a question like that, I'm going to do the shortcut first, and then I'm going to do the way that you're supposed to do it for CXC. All right? I'm going to do the shortcut first, and then I'm going to do the correct thing. So what I'm going to do now is what you're going to do on the multiple choice paper to answer the question. So draw your rectangle quickly. We're racing to draw the rectangle. All right, I am almost there. Last line connected. Draw the 2 horizontal line now. Hurry, we're in a race. Finish. 2 vertical lines now. 1, 2, finish. I'm sure you didn't beat me to that. I'm finished. Yeah, I'm finished. There it is. All right, draw the diagonal line now. Let me see if you can go quicker than I can. Finish. Beautiful. Everybody's finished. Cool. So now we're going to be talking about inheritance of sex. Now, we got pretty much XY, which represents the male. So we're looking at the gametes, we're looking at the genotype of the parent. So the parent, the father is going to be XY. Usually a X and a Y represents the male, and a X and the X represents the female. Now, when we are in genetic class, usually the girls tend to echo us a whole lot. They say that as boys, we are really hybrid girls. We are not real men, and we should give thanks for the mutation. So women have it to say that men are actually mutated women. And I'm going to show you why they say that. So I'm going to draw a big X. So that's a big X. So a woman is usually 2x. We soon get back to the Punnett square. Let's just share some genetic joke. So this is a woman, 2x. Now on the other side here, we have an x. We have an x and a half a x. Now look at the half a x, what is going to happen now? We are going to draw a line And now we get a man. See that? XY. So now we have a man. Here we have a woman. So the girls are saying that men are actually mutated women. It's because of mutation, the X never finished cross. And because of that, we should be saying thanks to them for being here. Even though the Bible, I think, already said that is our ribs make them. But when we get to genetics class, they want to change the whole thing to say that we are a mutated organism. Now you can tell me in the chat which it is. Is it that a man use his ribs to make a woman? Or is it that we are actually mutated women? The Y just never finished crossing. You can tell me in the chat. All right. So as you think about that, let us get on with the order of the day. Now, if we have this on an exam to solve, we're not going to take the entire day to solve it. So the first thing we write here is the genotype for the parent. So if you observe the letters there, representing genotype XX, XY. Now the phenotype, oh, this is genotype. So the genotype has to do with the genetic disposition, and we are using letters to represent the genotypic disposition. Then we have the phenotype. And this can be a little confusing for a lot of students because the phenotype here would have been male and female, but phenotype could also be about your height, your color, that type of thing, and we're going to be looking at a lot of that. We're looking at inheritance. So the phenotype for XY, anybody want to tell me what it is? What would be the phenotype for XY? If a genotype, what is the phenotype for XY? Man would make woman. All right, cool, not a problem. I see you're a Christian, not a problem. All right, so I should probably be telling you these stuff rather than heckling you. So for male, the phenotype would be male. The genotype would have been XY. So for the female, the phenotype there is going to be female or woman. Oh my, I'm messing up my paper. Ignore my messy paper. So you would have noticed here, I do hope that you're seeing well. I don't have the whiteboard. So you would have observed here, I have the phenotype, male, female, genotype, and this represents the letters, all right? And these letters will always change as we move along. And then I said that this belongs to the parent. Now I'm going to just jump and do a shortcut thing for the multiple choice, and then you'll be fine. Now, usually, I'm not being rude here. If there are kids watching, I'm not being rude. Usually, we use a little circle with an arrow being attached to the circle to represent the male. So this is the male. That little circle here with the arrow, that's the male. And he's on top. For whatever reason, I don't know. And then we have a female. Usually, she's at the bottom there. And it's just a circle with like a cross attached to it. Now, where you put the male or the female, it doesn't really matter. But we chose to put the female at the top. And we chose to put the male at the top in this case. So if the male is at the top, we are going to be putting the genotype for the male here and the phenotype for the female here. What I want you to help me with, what is it that I should be putting in these two boxes to know the F1 generation? Type it in the chat for me. What should I be putting in those boxes? Right, I want to know, we're doing inheritance of sex, and we're pretty much having the students go through it as a practical. So I'm doing it, they are doing it wherever they are, and they are walking me through the whole process as I am the amateur, right? And they are the teachers. So I am learning from them. All right, cool. So Barnett is saying a lot of things here. What about the other persons? What about the other persons? The question is for everyone, you know. All right. So Moore is saying we're going to put as genotype XX for female. Barnett is saying the same thing. Miller is saying the same thing. Powell is saying XY for male. Anybody else? Everybody need to take part. If you don't do the practical, it's like driving lessons. You can't stand up and watch a car drive and say that I'm a driver. You have to be a part. Don't be afraid to fail. We fail forward. All right, beautiful, 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 beautiful. So I'm going to take your advice. I'm going to put X and I'm going to put Y for the male. And you are going to do the same thing. X and Y for the male. All right. Now, if you don't remember these signs, just write the word, no? Male. And down here, so write female. Don't worry about not remembering the sign. Just write in any one of them, male or female. You don't really have to remember the sign, but it's good if you remember the sign. Now, on the other side for the female, they say I should put X and X. Now, this is like algebra that you have never done at school. It is pretty easy. Easy, cheesy, wheezy, beezy. All right, beautiful. Here we go. So here we have the allele, or as it were here, the chromosome, for the parent. Parent. XY here for daddy. XX. XX here for mommy. Now we are going to do the cross. Now wherever this side intercepts this side, we are going to put both letters. So that this, just let me draw two arrows here to show you. I'm going to be using a different color pen. So I'm going to show you with an arrow. That is going to go there. So that is going to go there. So you understand that they both intercept where you see the arrows. All right. So I'm going to be just writing it in as X, X. Beautiful. Look at that. So we deal with inheritance of sex. All right. Beautiful. So if this is going to be this, what are we going to put in this cell? Can you just tell me in the chat, what should we place in this cell? Come on, tell me in the chat. I really don't remember anything else, so you have to tell me in the chat. What are we going to put it? Where X and Y intercept in that cell, what should we put in that cell? Amoy saying XY. Is that true? I'm not sure if Miller was answering to. Oh, Miller is saying XY. Cheryl is saying XY. Anybody else? Anybody else want to be a part of this? Guys, remember you can't stay on the outside of a car and learn to drive. You got to get inside of a car to drive. All right, beautiful. So where they intercept, we're going to put X, Y. Now, for those persons who are going to be doing tertiary biology, please be reminded that this is monohybrid inheritance. It is pretty simple. But then you have dihybrid inheritance, and you might even get to trihybrid. But we're looking at just one trait being inherited, and that is why it is monohybrid inheritance. Beautiful. I think everybody is right. So now I wrote it in for you, and you're going to write it in your stuff for yourself. So now I'm going to look at where the X meet. I am assuming that we're supposed to have 2X where they intercept, and XY, that type of a thing. See that? Beautiful. Now listen to the question. We have just done the thing. The question is going to be, what will be the F1 generation, or the first filial generation? What will be the F1 generation for, we're looking at inheritance of sex. What will be the F1 generation for a man and a woman the next time they should conceive a child? What is the likely outcome? What the F1 generation will be? All right. So you have just solved that by doing the table here. But doing the table here is not enough. Now, you might be asked a question like this, and you're going to look at your thing. You might be asked to tell what percentage will be male, what percentage will be female. Now, tell me in the chat there what percentage will be male and what percentage will be female. Tell me in the chat. You know that yeah, you understand. Tell me in the chat of the F1 generation, or we call it first filial generation, and it's written like that. All right, so you'd have seen F1, mean first filial generation. All right, so it is going to be 50%, 50%, 50%, and that is going to be, might I just stop you here? As I see these responses coming in, I remember Dr. Medley on my exam paper, I wrote something like that. And Dr. Medley, who is the greatest in the world, says Mr. Wilson, Dr. Medley, out of the College of Agriculture. Big up yourself anywhere you are, Dr. Medley. You don't know your thing large. All right. Dr. Medley stopped me in my track, and you would have seen Amoy Mackenzie saying 50-50. But 50-50 what? You must say 50-50 what? So you will have to say 50% male, 50% female. To say 50-50, you haven't completed answering the question. And it's a good thing Dr. Medley started to mark me down for that. Or else probably I'll just say, oh yes, oh yes, oh yes. So like Moore would have said 50% male and 50% female. That is the correct way to express it. So you have expressed it as a percentage. Could you now express it as fraction for me, please? I want you to express the F1 generation as fraction. So that's percentage. I want fraction now. Express the same thing as fraction. Yeah, the syllabus could ask you to do anyone. Fraction, ratio, anyone. So everybody's nice saying 50-50%. As fraction, what will it be? Express the F1 generation as a fraction. Yes, you are doing nicely with percentage. Now I want fraction. Most person who have done well would have done well so far. Now I want fraction. So half the F1 generation will be male, while half the F1 generation will be female. All right, that's DD. Anybody else? Any other expression? Half female, half male. Remember that fraction here, we're going to go in the lowest term. So 15 to itself one time, 15 to 100 goes two times. So that is half, and of course half. So you want to make sure you do that maths, because we're testing if you know the maths. Sir, are you going to do this in HSB too? This is both biology and HSB. This class is both biology and HSB. We've been able to look at both syllabus at the same time. So if you're here, it's both biology and HSB. All right, remember that I did say that we can't just write half, we must write half male, half female. Very good. So now I want you to express it as a ratio. As a ratio. I remember that this is the simplest thing, right? We're going to be going on and getting a little more technical. All right, so now express it as a ratio for me, please. And then, having completed the inheritance of sex, and you understand that every time that a woman gets pregnant naturally, the chances of her having a male or a female will always be half and half, a 50-50%, or tell me what ratio. And that's a standard you want to remember when you get into the exam, should you see this on the multiple choice paper. How are we doing? All right. So it's going to be out. The ratio is going to be 1-to-1, because remember, in ratio, we are always going to be expressing it in its lowest form, right? So we can't say 2:2, right? We must say 2 into itself one time, 2 into itself one. So it's going to be 1-to-1. Always remember that the ratio is 1-to-1, and we remember we have to represent it as male and female. Beautiful. All right. So is there anybody in the room who still doesn't understand exactly what to do? All right, so what we did just now, that is for the multiple choice paper. All right, So in the multiple choice paper, we don't have to do any breakout of gametes and all of that. But when you solve, and it being paper 2 where you're asked to solve, the genotype that you have here for the parent, you'd have to break it out in gametes. And to break it out in gametes, all that you're going to do is to write these letters further apart, right? So you're going to have XY, and you're going to write gametes, right? So for the exam, if you have to do this for paper 2, you'll have to write this here as a breakout, XY, this is the gamete, XX, and XY, that's the gamete before you solve for the table, whether you are using the genetic diagram or the table. Now, can I just interest you a little more? I am going to be doing this inheritance again using the genetic diagram. All right? A lot of persons are afraid of this, but Mr. Wilson have to teach it. So let us go. So we're looking at the parent. So the first thing that we're going to write the phenotype. All right? So what is the phenotype of a parent? Please type in the chat. First thing we're going to write is the phenotype. What's the phenotype for the parent? We're still looking at inheritance of sex. What is the phenotype for the parent? Female, male, male, female. Thank you very much. That's wonderful. That's awesome. Now, what's the genotype? What's the genotype of a parent? The genetic representation, what do we use here for male and female? Come on, guys, don't fall asleep on me. I'm having a little headache and it's like really, really hot and humid. All right, so anybody else, the genotype? We're looking at the genotype now. You are going to be doing your cross now, you know. All right, beautiful, beautiful. Coming in a little slow, but beautiful. So the genotype for the male is going to be XY, and the genotype for the female is going to be XX. So that's the genotype for the parent. What is the next step? What is the next step? Having written the phenotype of a parent, the genotype of a parent, what is the next step? Tell me in the chat. What's the next step? What should I do next before I start the crossing? What else should I write? Anybody can tell me? So we have written the phenotype, we have written the genotype. What should we write now before we start the crossing? Give me a name. What should we write? Anybody remember? I'm looking for a G word. What should we write now? Anybody can tell me? All right, I'm looking for the word gamete. All right, very good. All right, so we have to write the gametes. A lot of persons tend to skip this out and move right into segregation, but we have to write the gametes. So we're just going to be spacing the letters out when we are writing the gamete. That's all that we need to do, just make them a little wider. So that's the gamete. All right, now this is the one that persons hate to do in the exam, the cross one. And that is why when I ended the Punnett Square just now, I showed you that after you have written the parental phenotype, the parental genotype, you are going to write the gametes. And then after you would have written the gametes, then you are going to use a table to do the cross. And the table would pretty much be segregation. So what we're going to be doing now is segregation with those lines crossing. Now, I like to start off by drawing one line down at a time. So I'm going to be drawing a straight line down under the first X. So this is my straight line. I hope that you are there. That's my first line. Now, my memory is not as good as anybody's memory, so I'm just going to write below it what it means. So for me, it means X. All right? So I carry down X right there. Now, we're going to be crossing the male with the female. All right? So we're going to be crossing across like that. So the next thing I'm going to do, I'm going to pull another line from the X on the other side And that is going to bring me a second X. All right? So that brings me a second X. All right? So 1 is already completed. Now, the other thing that I'm going to do, I'm working with the male, so I'm going to complete the male before I go on over to female. I'm going to pull a straight line down from the Y. And once I pull that straight line down, I'm going to place the Y at the end. So I place the Y at the end. Right? There we have it. A Y being placed at the end. Now, the other thing that we're going to do, we're now going to map it onto the female. So we're going to cross the other X onto this Y, and we're going to get XY. The naming convention usually put X before the Y. So there we go, we have XY. Now, if you observe here, this side is complete. Now, I don't want to become all confused in the exam with all the pressure, so I'm just going to draw a circle around it. And when I see the circle, it will tell me that there's no further work to be done right there. All right? Now we have to up and over to the other side. The other side has no straight lines. So what we're going to be doing, we're going to be doing the same thing. We're going to be pulling down a line. Here we have it. A line being drawn down. And once we do that, we're going to write the letter right below the line. Or the allele, one might say, or the chromosome as it is here. So write an X there. Now, this is the tricky part and where students now become confused. Now, this X is already connected to this Y. So you can't connect it to the Y a second time. So this X now must be connected to this X on the far side because it is not connected to that as yet. Remember, it's segregation and it's random. So now I'm going to go on over to the far side and I'm going to be drawing down that line, and we have a second X. Now, if you observe, now we have two alleles there, or two chromosomes there. So we are going to now be circling them, and the work there is finished. But we still have one that is incomplete. This is incomplete, this is incomplete, because it should have two lines coming from it, two lines coming from it, two lines ending, two lines ending, two lines ending. So we know that these must be connected. So what we're going to do again, remember we always start working with a straight line. So we're going to be drawing a straight line down again. And then at the bottom of this straight line, we're going to put an X again. So we would have done that. That's the X there. So now we realize that this here is finished because it has two lines. This is still incomplete. It needs another line. So what we're going to do, we're now just going to pull on another line all over, a line from Y, and we're now going to get XY. And you'll see here that it was completed nicely. Now, to label, here is going to be segregation, but the bottom here is your F1 generation. So this here would have been your offspring at the bottom here with the circle, and it's represented by F1. And this area here would have been random fertilization, or what we refer to as segregation. This area where the lines are being crossed. I do hope that everyone would have this now, and you are good. Probably you could just press print screen and it will be all saved, and you can go through the video again and work on that. Was that difficult? Tell me, how you feeling, guys? Was it good? Everything all right? Oh, lost right there. Oh, Powell. Oh, you got lost and I didn't see your message. So I'm going. So anybody else? Where's this lost? Anybody else? Tell me in the chat now. Don't be afraid. So I will just throw this paper. All right, I'll try to fold this paper. I'll fold this paper and I'll go again. Anybody else? Talk to me. I want to know what I'm doing. All right. No, you're not lost. Okay, good. The first thing you need to do when you are doing this cross so that you don't become confused is go one at a time. The rule again, or my rule, I want to use letters because it's letters you are going to use. You're going to say the letters. So I don't want to talk anything else but letters. All these letters must have 2 lines coming from them or 2 lines going to them, right? None of these letters should connect to one thing twice. It shouldn't connect to the same thing twice. So if you have X over here, you can't have X connecting to X over here so one time and X over here so 2 times. No, it can't work like that. All right, beautiful. But I don't understand Anderson, beautiful. But let me ask you before I get back to this cross. All right, did you understand the Punnett Square? When we did the Punnett Square just now, were you fine with that? Yes, no? Anderson, were you fine with the Punnett Square? Yeah, we could go to F2, but I'm not going to go to F2 today. I'm not going to go to F2 with this because this is pretty simple. I'm going to probably go to F2 when I'm looking at, when I go up a little notch now. I'm going to go to inheritance of dominance now. All right, beautiful. So let us just go through this one more time. Oh, Amar, you didn't understand. All right. Amar, you didn't understand? I didn't know. I thought you said that you understood. All right, never mind. I'll go through it again. So we're looking at the inheritance of 6. First thing we do, use the Punnett Square, which everybody understand or most persons understand. And if you go to my videos, you'll see them. But I understand that there are some students, the audiovisual reader don't help. They really need to see the teacher. So I decided that even though I sent you to watch those videos, I'm going to come on over here and do the cross again. All right, so let us do it again. So here it is. This man and this woman, they decide on having another child. They have already parented 99 girls. We want to know what the chances are of having a boy the next time they conceive. That's pretty much what we want to find out. So it is called in your text inheritance of sex or inheritance of dominance, and it pretty much comes on the exam paper anywhere between 45 and 60 on the multiple choice paper. Anywhere thereabout you see it. I think it's more 50 to 60. Thereabout you are going to see the genetic diagrams. You might see a question and you have to just draw up something quick, we can solve it. Beautiful. So we started out by writing the parental phenotype. Now the parental phenotype would have been male for the man. Phenotype is what you can see. And female for the lady. Having written that, next thing we wrote was the genotype. Now the genotype is the genetic disposition. So this was the phenotype. And now we are going to be writing the genotype. Now the genotype for the male, as default, we inherited this in our lessons. For male, it is represented by XY. So XY here for the male, XY there for the male. That's a genotype now. And for the female, it's going to be XX. All right? Normally, every teacher write that part on the board. Every teacher in the world write that part on the board. But the next part is the part that teachers tend to omit. And I'm saying that if your teacher is omitting it, just remind them, please, CXC require you to show the gametes. So the next thing we are going to write now is the gamete. All that we do when we are writing the gamete at your level is just to separate the X and the Y and separate the X and the X. So we just write an X over here and we write a Y over there. So let me just draw an arrow to show you what happened. All right. So the first X at the top, XY, that was actually the phenotype. Down the bottom where you say XY again with the arrows pointing to it, that is going to be the gamete. And we're going to do the same thing for the female, X over there, and you must label all areas. So you started out with parental phenotype, parental genotype, then we came down to the gametes, and now we are going to segregation, or what we call random fertilization. Now, this is where everybody becomes lost. And that is why when we're doing things like dihybrid or trihybrid inheritance, We don't use this diagram because by the time you finish drawing those lines, you are just going to have one big mess on the paper. We tend to use the Punnett square, but a much larger Punnett square. All right? Beautiful. So, in order to do this, I suggest that you start by drawing straight lines, one line at a time. So this is the first line. And when you draw this line, you are going to write that letter at the top, at the bottom. So now it's at the bottom. All right? So there I've written it at the bottom. Now the male on this side will have to cross to the female on this side because fertilization, so male and female must come together. So now I'm going to be crossing this X with the X from over by the female. So I just draw a line all over to the male here. And again, I write an X that comes over from the female. There it is, I drew it from all the way over the female to the male X. Now, this is finished because we have two lines coming. I'm seeing something else from the back, so I'm having a little problem. So we have two lines coming here, two letters here, that's finished. So just draw a little circle around it and say that, hey, that's enough now. Let's move right on. Then we're going to go down again and we're going to do the same thing for the Y. Draw a straight line down, draw the Y, there it is. Now we must attach that male to the female. So again, we're going to go over and we're going to draw from the other X, the one that doesn't have any line. We're going to draw from this X. We're going to draw a line down to the Y. And now we have XY again. Usually, X is written before the Y. Now, if you observe, we have completed the drawing on this side. So now we have to go over to the female. So I like to start from the far side. So I'm going to draw a line down. There we have it. Draw a line down. And then I'm going to place the same X at the top there at the bottom. Now, we have to now map this from the female, this X from the female, over with the male. But what we can't do, this X is already connected to, what is this? Connected to the Y here. So we can't map this to this Y again. We have to map it to the X on this side. All right, because you just go to one thing one time. What am I looking at? All right, beautiful. So I go there. This is already connected there. So I'm just going to be drawing from the other one that is on the far. And if you observe, we're using far and near. You'll observe that as you go through and make your observation. So now that is finished. But if you look at the one in the middle here, this and this, they are still incomplete because they have one line, while everything else would have two lines. If you observe, everything else have two lines, one line. So we know that these two must go together. So we just pull down another line from this X. Pull down another line. There's the line coming down straight. And then we're going to write the X at the base. So there we write the X at the end of it. Now we're going to now pull over a line now from the Y that we had there. And then anywhere the line goes, we're going to put the letter right at the end of the line. So again, we are going to have XY. So here we have the second XY. Right? So we end up now having XY, XX, XX. Was it any better? Was it any better, guys? Talk to me. Was it any clearer? Any clearer? Yes, no? Yes, no? Okay, it was beautiful. All right. One thing I can tell you, I am certain there's always a genetic question on the multiple choice. We're going to move up a little bit now. Now that you have mastered this, we're going to move up the complexity a notch higher. So we're going to look at inheritance of dominance. Dominance. I'm teaching you this lesson about the second or third time. There are some traits or some characteristics that are dominant. curly hair is dominant. And we looked at a long list when we did that long time lesson, long time last year. We're going to be looking at a chemical called PTC. It has a long fancy name, and I am not going to get to the long fancy name. But the thing about this chemical is that there are some persons in the population who can taste it, that it is bitter. And there are some persons in the population who cannot taste that it is bitter. The persons who can taste that it is bitter, it is considered that they have a dominant allele. Those who can't taste that it is bitter, they have the recessive allele. Now I'm going to give you the rule. And when you see the multiple choice question, you have to remember this rule. If you don't remember this rule, you just won't get it right. So the rule states that once the dominant allele is expressed, then the phenotype will also be displayed. A simple way to say it. So dominants, they are presented by, yes, usually by a capital letter. Yes. So once the dominant allele is expressed in the genotype, it is going to make itself present in the phenotype. Once it is expressed, whether it is expressed as heterozygous, and when we talk about heterozygous, we are talking about uppercase, as somebody said letter here, and I said that I was using letters. As when we talk about heterozygous, you have a capital letter and a common letter, right? But in genetics, what it would mean is like if you're a fat, then the alternate allele would have been slim, and then we spoke about black and white, that type of a thing. So the heterozygous here is that it is represented in the genotype with a capital letter and a common letter. The homozygous is represented by the same letters. However, you could be homozygous dominant or homozygous recessive. If you are homozygous recessive, you are going to have two common letters. And if you are homozygous dominant, you are going to have two capital letters. Oh my. Hello, guys. Did I give you too much? All right, let me slow it down. Not a problem. Somebody type for me. Homozygous. Powell would have typed something there. Capital A and common A. That's heterozygous. Homozygous. You're seeing two common A and two capital A. That is homozygous. But the two common A is homozygous recessive. And remember that we could use any letter. And the two capital A is homozygous dominant. Don't you ever say, don't you ever be mistaken that dominance mean that the allele, don't you ever mistake that dominance mean that it is the popular one. Dominance has nothing to do with popularity. Dominance might be among popularity, but it has nothing to do with popularity. And most time when we're giving a definition and making an easy explanation for dominance, we say the trait that is the good trait. Hey, 10 finger or 10 fingers is recessive. The dominant allele is 12. So this is one example where the recessiveness is nice, or you enjoy the recessiveness. Because some persons really don't want to have 12 fingers. They want to have 10 because 10 is popular. But 10 is pretty much a recessive allele. While 12 is the dominant allele. All right? So that said, and I just allele up your mind and your brains and all of that. And some person said, hey, you know what? I'm not going any further with Mr. Wilson and this genetics thing. I am just packing up and just taking the easy way out. Don't pack up. Don't take the easy way out. Just go with us and we will slow down to teach you the thing. All right? We are in no rush. We'll slow down to teach you. All right. So let me just go with that rule again. we're saying when with inheritance of dominance, once the allele is represented in the genotype, it will also make itself present or popular among the phenotype. So once any at all you see it in the genotype, that is what is going to be displayed in the phenotype. Just let me give you an example. Suppose we were working, I'm going back for a piece of paper. Suppose we were working with T for tall, and usually we define the allele with what means what. So this capital T here means tall, and T will represent the genotype. And a common T would be the recessive allele. Now, if we were supposed to show you this, would this person be tall or short? Remember, capital T is dominant. Would this person, this heterozygous individual, would this person be tall or short? Type in the chat for me, tall or short, whichever one. So am I saying that this person is going to be tall? Anybody else? Anybody else? All right, good. So this individual is definitely going to be tall, and the person is going to be tall because the dominant allele is a part of the genotype. This is the dominant allele. Now tell me about this individual, this second individual. Will this person be tall or short? Will this person be tall or short? Yeah, the first person was heterozygous tall. Good. So this person is going to be tall. Beautiful. So when you look at it, in both cases, the dominant allele is expressed, and as such, both persons are tall. However, oh, can I just fold this here? However, if the person had these, would this person be tall or short? Applying the rule of inheritance of dominance, would this person be tall or short? So I'm taking my answers from more downward. Would this person be tall or short? Two homozygous recessive. The person is going to be short. Beautiful. Now, if this wasn't a guess game, it would indicate to me that you understand the rules. Remember, once the dominant allele is expressed, it will make itself present and dominant in the phenotype. So you're going to see it in the phenotype. All right? Good. Understand? Beautiful. Wonderful. I just set you guys up to be on your own because now I'm going to start talking genetic language. So to answer a question like this on the paper too, the first thing you have to do is to define your alleles. So you are going to let capital T be the dominant allele. Right? Capital T be the dominant allele, while common T will be the recessive. So that is the first thing you're going to do. Define your allele. Capital T dot dot. Now you don't have to write long statement like this. You could probably just write a capital T, and you could write tall, and you could write a common T, and you could write short. Right? But in most cases, you are not going to remember that what that means. So it's nice if you write out the statement. Now, after you would have done that, in this case, we really don't require male or female. We're not going to be talking anything about an XX now. What we're going to be talking about now is T's, right? So we're saying that persons who can taste PTC, they have the dominant allele. Persons who can't taste PTC, they have the recessive allele. Can I just show something on you without warning? Here we have a man and a woman. They are both heterozygous for the trait to taste PTC. Tell me what the F1 generation will be. I hope I didn't show this one way over your head. So there are two persons who are heterozygous. And when they are heterozygous, it means that they have uppercase and lowercase letters. I am using letters for those persons who are not here as yet, meaning that you haven't locked onto the thing as yet. So there are two persons, a male and a female. We really don't business about X and Y now. We're not really treating with that. All right. We're treating with the T's. So one of the parent has a, they are, okay, let me repeat. We have two parents. They are both heterozygous for the trait to taste PTC. Tell me or show me what the genotype of the F1 generation will be. So you're going to write the genotype of the F1 generation. And just let me just do my calculation here. Or, oh my, there are some persons who are out of the block really, really fast. Remember to put commas between those, put commas between those. All right, so I am not good like more, so I'm going to be just drawing up my thing. I want to do it as fast as more, so they are both heterozygous. I'm going to do like more very quickly, like more. More is the guru, it seems, and I know that where they intercept, we're going to have that. So let me see what more did here. No more. No more. More, you would have to go back to the drawing board. No more. No more. Use the shortcut method for me now. Just show me in the Punnett Square. Oh, use the shortcut method. No Powell. No Powell. Why it look like I am the person who's getting it wrong? Let me say F1 generation. What does that mean? No, 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 no. All right, so probably you didn't understand what I said. So I said that both parents are heterozygous. I am going to give you the allele for the parent. So the parent would be, one parent would be that, and the other parent is going to be that. So there you have the allele for both parents. What is going to be, and that's heterozygous, right? So I am having problem, persons are not getting it right. Those offspring, no. So we're going to have, all right, Barnett, come back and come back real strong. Why make you have so much attitude, Barnett? Barnett, why you come back with so much attitude? All right, come on, guys. Work it out for me. So all I asked for now is just for the F1 generation. I didn't ask for anything else but the F1 generation. So what we have here is two heterozygous parents for PTC. And now I'd ask you, what will be the genotype of the F1 generation? So we are crossing those two things that are placed there as heterozygous, and we want to see how best we can get the answers. All right? And if I can help you, what Barnett has there, that is correct. So work it until you get that. If you don't get that, let us know so we can assist you. All right, so we're looking at that. There are other questions I'm going to be asking you about that. So my phone. Oh, your phone changed it the first time. Okay, I see. Sorry. All right, so So Didi, you actually got it. I don't know why it keep flogging. YouTube is saying no. All right, all right. So I say everybody's showing me that, but everybody getting it. All right. I need to wait on other persons. Come on, guys. I want everybody to get the practice. All right. Never mind. If you don't get it this time, you'll get it the other time around. I don't think we can teach anything else this evening. All right. Look at what we have. That is the result there. Remember where the intercept? We have common T and capital T, common T. And then we have here capital T, common T, and remember where the intercept. So that's how we came up with that answer. All right, I was doing it pretty fast. I'm very sorry for the handwriting. I was racing. I was playing around. All right, so I use the chart. Which of the chart? The line stuff. I am the Punnett Square guy. You give me anything like this to do, I'm doing it in a Punnett Square. I ain't drawing in the lines. I wonder which one to draw. No, I'm not doing that. All right. But remember, you have to do segregation and write the gametes and all of that when you are doing it for paper 2. All right. Beautiful. So you have solved that. Some persons didn't get it. I'll just show you my answer again, and then I'm going to give you another one to do. And this time, what we're going to be doing, we're going to be crossing. We're still working with inheritance of dominance. So we're now going to be crossing homozygous dominant parent with a heterozygous parent. Cross and tell me. Oh, you use a Punnett square. Okay, I want you now to cross. Did I say homozygous dominant parent with a heterozygous parent? Oh no! Don't go there yet. Mr. Wilson's not being nice. Mr. Wilson's being naughty. All right, here. Oh, that's fine. Here, what I want you to do. Let's finish this question. Now, I want you to tell me the phenotypic disposition of the offspring. The offspring here, tell me the phenotypic disposition. You could tell me the phenotypic disposition in ratio, in fraction, in percentage. What is the phenotypic disposition? So I'm seeing the genotype now. Tell me the phenotypic disposition. Phenotypic disposition. What will the phenotype be? And what number you attach to them, that type of a thing. I know. We're going to bring the curtain down on pretty much inheritance of dominance. And when I get here on Sunday, I will be looking at inheritance of recessiveness. And I am going to be looking at either inheritance of codominance and incomplete dominance. All right, we have recessiveness. We have incomplete dominance. Just let me look at my plate of things to do. We have recessiveness, where we're going to be looking at albinism and cystic fibrosis. And then we have another color blindness, inheritance of sex-linked inheritance. This is sex-linked inheritance, where we're going to be having, come on, little old head. We're going to be looking at hemophilia and cystic fibrosis, which is, of course, sex-linked inheritance. We're going to be looking at that. Now, when we look at sex-linked inheritance, it gets a little more technical. And when we look at codominance, it becomes a little more technical. So what I'm going to do, after dominance, I'm going to be looking at recessiveness, right? And that should be a little easier. And then you will make the graduation until we get a little more complex, that type of a thing. Now, remember here, we're not talking about tall and short here. We're talking about taste. We're talking about PTC, the chemical PTC. So why person saying so? So you're saying 50% will be taster. I would have said. What about the others? Is it 50% that would be taster? I don't think that is correct. No, it's not 50%, it's more than 50%. Remember the law. The law says once the dominant allele makes representation in the genotype, then the phenotypic disposition will be that of the dominant allele. So if we had homozygous T, heterozygous T, and homozygous recessive T, then it can't be 100% tall. It can't be 100% taster. No. No, 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 no. Look at what you have there. Only the dominant T's are tasters, you know. Only where you see the dominant T expressed, we have taster. If there's no dominant T there, then they can't taste. So yes, it's going to be 75% tasters, but you can't just say 75% taster. You need to talk about the other part as well. So if you say 75% taster, then it's going to be 25% non-taster. You must make representation of both, unless it's on the multiple choice paper, or unless the question definitely asks just to represent that fact. All right. So no, not 50. No, no. What will so it will be 50% heterozygous, 25% homozygous dominant, and of course 25% homozygous recessive. But you have to make sure that you are paying attention to the question that is being asked, or else you are going to work it out and still get it wrong. Right? What is it that they want you to answer? Is it that they want you to answer to ratio? Is it that they want you to answer to percentage? Is it that they just need the phenotype? What is it that they want you to answer to? You need to make sure that you have that all locked and concretized in your head so that you won't make that mistake when you get to the exam. I think we have been doing well so far. So we're going to be crossing two persons now, and they are both homozygous recessive. I don't want you to do any cross. I want you to tell me the answer now. Go quickly. Two homozygous recessive individual cross. What will be the F1? What will be the phenotype of the F1 generation? We're crossing two homozygous recessive individuals. What will be the F1, the phenotype of the F1 generation? Two homozygous recessive. Come on, guys, this one is easy. Come on, why don't do any cross? Just answer. If you have to work it out, work it out. But you really don't have to work it out. Amoy, you are giving me genotype. I'm asking for the phenotypic disposition. More, we are dealing with taste, not height. DD, you can't just say non-taster. You have to say how many. Is it that all? Is it one? Powell, you are giving me genotype, but I'm asking for phenotype. Anderson, you are giving me genotype, but I'm asking for phenotype. For non-taster of PTC. Thank you very much, more. That's what it's going to be. 100% non-taster. Very well, Powell. That's what it should be. Everything will be the recessive allele. So none of these persons will be able to taste PTC. All right, you did well there. Let me give you another one for those persons who still don't get it. Now you're crossing two 100% homozygous recessive. You're not answering the question, am I? We're talking about taster and non-taster. So you have to answer and give me the answer as taster and non-taster. All right? All right, let's look at this one. We are crossing two homozygous dominant individual for PTC. what is going to be the phenotypic disposition of the F1 generation? Phenotypic disposition of the F1 generation? All right, so with this question, we're going to be bringing the curtain down this evening. We look forward to seeing you come, wow, I look forward to see you come Sunday. There is something else I just wanted to read out for you guys. I'm going to bring the curtain down on this. If you didn't have your question doing, I'd ask you to guess it. But being that you have your question doing there, I am going to be sharing something with you. Oh, what was I saying? I was saying, Amoy, that it should be a 100% taster, right? Because we're talking about the phenotypic disposition. So we're asking about what characteristic or what trait will be inherited. Remember, we're doing inheritance. So we ask what the phenotypic disposition will be. You have to tell us about the trait. All right. And the trait in this case is, of course, going to be whether the person can taste or can't taste. Now, the other question that I gave was that we had two parents and they were both homozygous dominant. And we wanted to know what the F1 generation will be. And Barnett is saying that they're going to be 100% taster. So everybody in that family will be able to taste that bitter thing. Is it possible for a person iris color to change? That's a big one. That's a big one. I have seen persons with two different iris, but as to if the color can change, I'm not too certain. I'm not too certain. I don't know if you're talking about cataract or anything like that. Are you talking about something like that, like a disease? Or are you just talking about today the eye is blue and tomorrow it is red? With cataract, I know it can change. Or with the eye disease, I'm sure that it can change. But as to change naturally and nothing is wrong with you, today your iris is blue and tomorrow it is pink or purple. I don't know if the eye works like that. I really don't know. I've never seen anything or had any literature like that. But if you do, you could share it with me. I'll be too happy to read. All right, beautiful. Somebody said, sir, question. Oh, so Paul was asking the question. Okay, I got a copy of the 2021 HSB paper. I got it today. Some good Samaritan sent it to us. Yes, and I have the 2021 bio paper as well. Right? So we're going to be working on it. But before I leave you, I'm going to tell you about some of the questions that was on the paper if you didn't see the paper. I did suggest some time ago that you should make sure that you complete the 2016 paper. And I'd ask you to complete the 2016 multiple choice paper because when I looked at last year paper, it was a repeat of most of the questions from 2015. So because of that, I thought that it would have been prudent for this year you concentrate a whole lot on the 2016 paper. I think that the 2016 paper was a little challenging, so you want to make sure that you do that and more. Look at those questions. Some of those questions might be repeated. I don't know. I don't work with CXD. It's just an assumption. The multiple choice, the paper two for the 2020, 2021 we're in. I got a copy today. I could just tell you some of the things. I was a little baffled just to say as to some stuff I saw on it, and it was a little frightening. The paper started off with two diagrams, and the diagrams were of a respiratory system, looking at breathing. Then it went down, and the other part looked at the alveoli. And the question, I'm just looking at the diagrams, and then there was something there on the currency of the cell with ATP and all of that. And this came on some paper as multiple choice. ADP to ADP, that thing, it was there for question number one as well. And then when we got down in question number 1. I don't know if you remember that graph that we spoke about vital capacity and all of that. And I told you that you needed to have that graph. And that was on the paper as well. I'm just doing diagrams. I'm not doing everything. I'm up and through. And then, interestingly, this topic that we did not look at, the lymphatic system was on the paper. We haven't looked at that yet. I think it's asking us to identify the lymph duct and the lymph node. We were supposed to identify those on the paper, and we were supposed to state one function of the spleen. One function of the spleen. That stood out because the spleen. All right. And then after we would have looked at that, then came down, let me just make sure I'm seeing this diagram well. Blood flow from the artery to the vein. I think this should be a capillary bed. I think it is a capillary bed, some diagram there. I'm not going to go through the question. I'm just looking at some diagrams. There was a micrograph or picture of cells. Yeah, here it comes up. And to me, Mr. Wilson, I don't know if it's because it was, I don't know if this was photocopy or what, but it was sent to me via PDF. To me, I don't know if the picture quality is the best. Persons are going to be having a little challenge to look at it. I think one of the cell there is the cheek cell. I'm wondering if the other one is the muscle cell because I would be looking for striation or something like that. I don't know. I am looking at it, and yeah. So that wasn't so much of a nice photo for me. It wasn't so much of a nice photo. Probably if I stop and look at it a little more, I would probably be better able to identify. Then there's a nice table here that says complete the table below by naming the correct cell structure for each scenario. The first row has been completed for you. On the first row, they said mitochondria. And then it says Dr. One has observed that his host plant is wilt. Which cell organelle is most likely impacted? All right, so this is stepping up the thing a notch. I know that you were expecting the difference between plant cell and animal cell, but what structure in the plant cell? It is which structure in the did I ask which cell organelle is most likely impacted? All right, so there we are looking at what the cytoplasm, that is where we would have most of the liquid things. So that you have to know the function of these in order to answer. It says Aaron hopes to remove carbon dioxide from the air by stimulating the growth of a plant. Which cell organelle would be important in reducing carbon dioxide level? Now, which cell organelle would be important? And it's a cellular, not we're not talking about the stomata. No, it's a which cell organelle would be important in reducing carbon dioxide level. So you see there, you have to understand the functions of the cell in order. And then comes up another sunflower picture. I guess you'll see it as soon as I vlog. I will vlog on them. AIDS is on the paper again. AIDS is, remember when I looked at that statistic thing, AIDS is like it has a permanent seat on the paper. All right, and then we come up now, we have methods of birth control. That's the other question on the paper. And then, I see countrywide, whatever. I won't go into that. That's still birth control. And then here we have balanced diet in section B, question 5 comes up with diet. And then same diet, a big on diet. And then water purification, around question 6, water purification. And then we have culture. This is popular on the multiple choice paper. We haven't seen it on paper 2 for a while, but it is now on paper 2. Agar plates at the end of the experiment there being conducted. We have not done that experiment on the channel, but we are definitely going to be looking at that. And that pretty much pulls us to the end of the exam paper there. The paper will be available in some format as I am guided. I like to ensure that I take the instruction from the persons who usually send me these stuff. So it is there, and we're going to make it available in some way, shape, or form. The paper is not so walk over. The quality of the questions, even those that we have come so accustomed to know, those have changed. 'Cause if you look at the cells, the micrograph, I mean, it's most students not looking for that. We're looking for the regular draw cell. And if we look at the plant cell versus the animal cell, that cell structure table, it is pretty much out of what we were looking for. We are accustomed to seeing what organelle is present and not present, but it has changed a little bit. Now, I did say that the 2016 paper was a little challenging. And if last year paper was so excited, I am thinking that this year paper might not be as excited. So I want to ensure that we get everything in. Yes, we're going to be doing the paper twos for the persons for HSB because we're having a consumption at HSB. So we're going to be doing some HSB paper too. We've started some, but I have so much stuff doing. Forgive me. I sometimes have to just do some short stuff to get things online until I have time to do the long stuff. I say I am occupied with something in my brain. I'm playing with the stapler. All right, so that's going to be for the show today. Remember, it is, of course, Mr. Wilson Live, and we are streaming. We're streaming from Kingston, Jamaica, and we stream 5:05 on a Wednesday, Friday, and Sunday. If you are not yet a subscriber or a member of this family, please use the time to do so now. Remember to like, share, and subscribe. And when you subscribe, remember to hit that notification bell and select all so that you will be notified as soon as there is a new publication or whenever there's a new publication. And the next little thing, remember to give the show some love. And I'm sure you know what love is. Give the show some love. All right, take care until Sunday. I'll be traveling on Sunday, so if I get in a little late, just forgive me. I'm going to be traveling. All right. So take care. All the best for the weekend. Be safe. Remember to study and practice. Good evening. All right. My pleasure.